In this video, I'll be focusing on the very first steps of pregnancy: fertilization, where sperm meets egg, and implantation, when the early embryo embeds itself in the wall of the uterus. Throughout our discussion of pregnancy, we're really going to be focusing on selected topics. We'll talk about fertilization and implantation, we'll talk about the placenta, we'll talk about labor and delivery and lactation, but we're not going to go into a lot of detail on embryonic development. What you're looking at here is the moment of fertilization. You have an egg, an oocyte, surrounded by spermatozoa. But before you get to this point, the sperm need to become activated, and that's a process called capacitation. The capacitation of sperm is a two-step process. The first is an increase in the sperm's motility. Basically, when the flagellum of the sperm cell begins beating. This happens in the male when the spermatozoa encounter seminal fluid in the ejaculatory duct. So the sperm are formed in the seminiferous tubules, they're stored in the epididymis. When they move through the vas deferens up to the ejaculatory duct, they actually aren't moving, their flagella aren't beating. But once they mix with seminal fluid, then they start beating their flagella, and they become capable of moving on their own. So that's the first step in capacitation. The second step in capacitation is a weakening of the cell membrane of the sperm. And this makes it easier for the sperm to dump its acrosomal enzymes on the structures that surround and protect the oocytes. And this actually happens once the sperm enters the female reproductive tract. There's a molecule that inhibits capacitation that's produced by the male, and that molecule gets diluted out once semen enters the female reproductive tract. Now, the second part of capacitation, making it easier to release the acrosomal enzymes, is necessary because the oocyte is actually protected by a couple of surrounding structures. You have the corona radiata, and the corona radiata are the follicular granulosa cells that surround the oocyte. You can't really see too many on here, but if I could sketch in where they are, they'd basically be this halo of granulosa cells around the oocyte. So sperm cells need to be able to penetrate that halo and penetrate between those cells in order to gain access to the oocytes. The other structure that the spermatozoa need to penetrate is the zona pellucida. And the zona pellucida is this thick glycoprotein coat around the oocyte. If you're looking on here, this structure is the zona pellucida. It's if the oocyte is the yolk, the zona pellucida is the white around it. So it's glycoproteins. It's made up of protein with some carbohydrate molecules attached to it. It's produced by both the oocyte and the granulosa cells. Some of the proteins in the zona pellucida act as receptors for the sperm to bind, so the sperm can kind of latch onto the zona pellucida. And it's almost sort of like a soft shell around the oocyte. And in fact, when the fertilized ovum loses the zona pellucida before implanting in the uterine wall, that process is called hatching, even though it's not quite the same process as, say, a chicken hatching out of its egg. Now, there are multiple steps in the process of fertilization, and this slide walks you through each of those steps. You have the oocyte here, you have the zona pellucida here, and you have the corona radiata here. So first, the sperm cell needs to get past the corona radiata. And this is helped by an enzyme that's present on the surface of the sperm called hyaluronidase. Hyaluronidase breaks down hyaluronin, which is part of the matrix of the corona radiata. So it kind of burrows in between the cells, separating them. Now, once the sperm cells get through the corona radiata, they encounter the zona pellucida. And as I said, the zona pellucida has receptors for sperm cells. Once the sperm binds to these receptors, that causes an increase in calcium in the sperm cell, which triggers something called the acrosomal reaction. And the acrosomal reaction is just a breakdown of the plasma membrane and the acrosomal membranes so that the acrosomal contents, those enzymes in the acrosome, get released onto the zona pellucida and can kind of eat through it, enzymes that will break down the zona pellucida. So in step 3, those acrosomal enzymes eat through the zona pellucida, creating a pathway to the oocyte. Now, the zona pellucida surrounds the entire egg. It takes hundreds of sperm cells, at least, to actually get through the zona pellucida. One sperm cannot carry enough acrosomal enzymes to do it on its own. So even though it's only one sperm that actually delivers its genetic material to the oocyte, it takes hundreds to get a single sperm to the oocyte. But once that sperm does make it through, it binds to receptors on the surface of the oocyte. The plasma membrane of the sperm and the oocyte fuse so that the nucleus in the head of the sperm enters into the oocyte. Once the sperm nucleus does get into the oocyte cytoplasm, that triggers a rise in calcium levels that sets off the cortical reaction. And in the cortical reaction, you get exocytosis of these preformed granules that are sitting there just under the surface, just under the plasma membrane of the oocyte. And once the sperm nucleus goes in, these vesicles are released, and they dump their contents all over, completely around the surface of the oocyte. And there are two main effects of this cortical reaction. You get a hardening of the zona pellucida, and you get clipping off of the sperm receptors. So basically, what this does is this makes it so that you only get one sperm fertilizing that oocyte. That's what you need to make a viable embryo. If you had multiple sperm nuclei entering into that oocyte, it wouldn't be viable. You'd have too many chromosomes, you'd have too many genes. That would not be a viable embryo. So, as soon as one sperm makes it through into the oocyte locks everyone else out, hardens the zona pellucida, cuts off those sperm receptors, nobody else gets in. This figure summarizes the very earliest stages of embryonic development. This first box here is basically the last slide. So fertilization, the very first step in creating an embryo. Fertilization triggers the oocyte to complete meiosis, and you get formation of the second polar body. You also get a bit of reorganization and kind of some minor structural changes in the nuclei of both the oocyte and the sperm cell, forming the female pronucleus and the male pronucleus. As these pronuclei, male and female, are forming, the spindle fibers start to form, setting the cell up to begin cell division. Before that can happen, you have amphimixis. The pronuclei they lose their membranes and the genetic material intermixes. So amphimixis is basically the fusion of the two pronuclei, where you go from two haploid nuclei to one full diploid set of chromosomes. The first few cell divisions are referred to as cleavage because they happen very, very rapidly, and the ovum, the fertilized oocyte, is now called an ovum. It basically divides and divides and divides rapidly without growing in size. The cells don't actually increase in size. So what you end up with is numerous small cells still contained at this point within the zona pellucida. This cleavage stage continues, just dividing over and over and over until implantation. This figure goes through the steps involved in implantation of an embryo into the uterine wall. The embryo, or to be more technical, the zygote at this stage is referred to as a blastocyst. Zygote is the earliest stage of development, and then you have the embryo, and then later on you have the fetus. The zygote at this stage is referred to as a blastocyst. The term cyst refers to a fluid-filled cavity. And the reason why this is called a blastocyst is because you have a fluid center that's contained by a layer of cells. This layer of cells is called the trophoblast. This trophoblast is what secretes human chorionic gonadotropin that can be detected early on in pregnancy in the blood and urine of a pregnant woman. You also have to one side of this fluid-filled cavity, you have the inner cell mass, and it's this mass of cells that develop into the fetus and eventually become the baby. Now, implantation usually occurs about a week after fertilization. There are ovarian hormones, progesterone primarily produced by the corpus luteum, that prepare the endometrium for implantation, that make sure that it is well developed, that the uterine glands are producing the glycogen the zygote needs in order to fuel development. It's a complex process. The endometrium has to produce the right hormones and the right growth factors to promote attachment and invasion by the embryo. The majority of pregnancy losses occur before or during implantation. It's really not all that uncommon for an embryo to not make a successful implantation. During a successful implantation, the embryo basically digs its way into the endometrium of the uterus. The trophoblast, cells of the trophoblast, secrete enzymes that break down the connective tissue between the uterine epithelial cells so that the trophoblast itself can kind of invade into that wall. Then the uterine epithelium of the mother basically repairs itself and builds a structure over the embryo. So the embryo, once it's successfully implanted, is completely surrounded by endometrial tissue. So you have maternal tissue there, maternal tissue surrounding the embryo there, and the embryo itself develops here, completely embedded within the uterine wall, out of contact with the uterine cavity. This is where the embryo will continue to grow and develop through the next 9 months. After spending some time studying this video, you should be able to address these first two questions in particular. Talk about the processes of fertilization, and talk about what implantation is. I will say implantation almost always occurs in the uterus. If implantation occurs anywhere other than the uterus, it's referred to as an ectopic pregnancy. And if it occurs in the fallopian tubes, which really don't have space for a developing fetus, it can be very dangerous for the mother. It's not a viable pregnancy for the child, and if it's not taken out, it can kill the mother as well. Ectopic pregnancies in other locations that do have some space for the fetus to grow are still very, very dangerous for both mother and child. Like in the abdominal cavity, I mean, they're still very dangerous, but they have some potential to be viable with careful medical supervision. All right. So you should also be able to discuss some terminology. You should be able to talk about what the corona radiata and the zona pellucida are. You should be able to discuss the process of capacitation, what it means for the sperm, what it does to the sperm, and amphimixis, what amphimixis is.